Hi, I'm Sarah Witzke and I'm a Senior Application Scientist here at Chemical Computing Group. Today, I'm going to show you template-based docking in Mo. We use template-based docking to match a congeneric series of molecules into a protein pocket. In the template-based docking method, we match the dock ligands against the so-called template, such as the crystallographic ligand. This will not only save us time during the placement, but it will also ensure that we obtain relevant binding poses similar to the known binding mode. Defining how to match the template can be done in several ways. You can either select atoms in the Mo interface, you can use a sketcher or a smarts pattern. Both of these will allow you to use wildcards as well. You can also match automatically using the so-called similarity mode rather than an exact atom-to-atom correspondence. So let's get started. I'm going to use our open panel to navigate to $mo sample mole, our directory of example files. Here I'll start typing the file name and the panel will automatically jump and I can find 1RO6.pdb. This particular crystal structure holds two copies of the same protein. So I'll use the symmetry option called biomolecule assembly to load in the biologically relevant state. For this protein, that's the monomer. Now what I've opened here is the phosphodiesterase 4 enzyme, or PDE4. There's also a bound inhibitor about here in the center called rolipram. In a minute, I'll come back to how we can take a closer look at the binding pocket. However, before we do any kind of modeling, and that includes docking, we need to prepare this raw crystal structure for common issues such as missing atoms. We need to set protonation states and deal with strain and so on. For that, we're going to use the QuickPrep tool. So QuickPrep is our three-step preparation protocol. Step 1 deals with missing atoms and setting protonation states. Step 2 prepares the structure for modeling with tethers and restraints. And step 3 runs an energy refinement to relieve strain and clashes. I'll start this with defaults. And you'll note that as soon as a calculation is running, the top left corner of the Mo window will explain what it's doing and how far it is. When this text disappears, it means the calculation has finished. Let's take a closer look at QuickPrep. So step 1 is looking through the structure and identifying any issues. And when it identifies missing atoms, it will add in missing side chain atoms and smaller loops. It will then move on to add hydrogens. It will do it with our algorithm called Protonate 3D. Protonate 3D will look at ionization states, tautomeric states, but also at rotameric positions of the hydrogens. So at the end, we end up with the most optimal hydrogen bonding network. Then we'll move down to step number 2, preparing the structure for modeling. Here we'll add a so-called tether to the receptor atoms. This will allow them to move a bit, but not change their crystallographic position too much. We will fix any atoms that are more than 8 angstroms away from the ligand. This step will save time when we reach the third step, which is the energy refinement or minimization. Here we will allow the atoms to move a bit to relieve any kind of clashes and strain that we might have. Let me close the panel again. I can see we are at the last energy minimization step. Let's focus on the binding pocket by using the side view option. Side view will automatically detect the ligand and zoom in on it. It will also render the ligand in a slightly thicker stick mode, while the nearby residues are in a thinner line mode. I like to change the color of my ligand, so I'm going to open the System Manager and attach it to the right-hand side of the Mo window. In my System Manager, I have an overview of what's loaded, and I can work individually on receptor atoms, solvent atoms, and ligand atoms. Here, I'm going to hide the ribbons of the protein, and I'm going to color the ligand orange. Now, I'm used to looking at this ligand in a very specific rotation, so let me just rotate it like this and show you what we have here. Now, this ligand, this inhibitor here, is called rolipram. And rolipram has a scaffold like many PDE4 inhibitors consisting of a so-called catechol moiety. This is a 6-membered aromatic ring with 2 oxygens on. In the case of rolipram, I have a couple of different R groups on the left-hand side of the scaffold, and I have an R group on the right-hand side consisting of a lactam ring you see here. Now, Mo will automatically detect and display hydrogen bonds as dotted lines. I can see that I have a very important hydrogen bond going from one of the catechol oxygens down to a conserved glutamine. So this roliprem inhibitor displays a very common binding mode of PDE4 inhibitors. I have the scaffold placed next to the glutamine, and I have a hydrogen bond displayed. On the other end of my ligand, so here at the lactam ring, I also have hydrogen bonds to the receptor. Now, I have a data set of similar ligands, so congeneric ligands, around the same scaffold as roliprem, but with different R groups. I'm interested in investigating how they will bind to the PDE4 when I change the R groups. In other words, will the binding mode be maintained, and how will my R groups interact with the receptor? And that's exactly what I'm going to use template-based docking for. So I'm going to the header to compute and down to dock. This opens our unified dock panel. Over on the left, we have all the different docking scenarios. We have a general small molecule docking. We have the template-based docking we'll do today, docking into experimental electron density, reaction-based covalent docking, protein-protein docking, and interfaces to external docking tools. Now have a look while I go back through these different scenarios. What you'll note, it's only this middle part of the panel that changes. The rest of the panel stays the same, independent of the kind of docking that we do. I'll make sure we end up at template. Now let's focus on the main part of the panel. This is split into 3 sections. Up at the top we define the receptor, and here we can also use a database of different protein structures for ensemble docking. In the middle we define the ligand and the placement method, while at the bottom we define the refinement options and the scoring options. So with any panel in Mo, we start at the top, we work our way down, and when we reach the bottom, we have all the settings that we need. I'm going to move the dock panel to the side, and then we can set it up. Now for the receptor, I'll use the one in Mo, and for the site, I'll use where the current ligand is. Today, I won't be using a pharmacophore density. For the ligand, I'll browse and navigate back into our example directory, where I can find 1R06_ligands, my database of ligands of interest. Then I need to decide the placement method. Here I'll switch to substructure, and I need to define a template. The panel guides me and suggests me to use selected atoms, which is exactly what I'll be doing. I'll go into Mo, and I'll use the 2D overlay, and here I'll select the catechol scaffold. Let me hide the 2D overview and continue with the dock panel. I'll move down, I'll leave refinement on rigid receptor and my default scoring on our generalized Bourne implicit solvent scoring function. The only thing I'll do is, in the interest of time, I'm going to turn down the number of return poses to 3. Finally, I'm going to add an underscore template to my output name so that I remind myself which method I've been using. When I press run, the results be written to a database, which automatically is opened up in this database viewer you see. We'll take a closer look at this in just a minute, but before we do so, let's go and have a closer look at the dock panel. So for the receptor, we're using the one in Mo. For the calculation, we're currently including both receptor and solvent atoms. If I was interested in running this calculation without these solvent atoms present, I don't need to delete them. I can simply change the option to just include the receptor atoms in the docking calculation. Moving down to the ligands, here I'm using ligands in a mode database file, but I could also use an SD file or, for instance, directly from a sketcher. The way I'm placing them in the pocket is by superposing them on a common substructure. Here my substructure is defined by selected atoms. I could actually have multiple cores in the Mo window and select multiple cores so that I could dock different scaffolds. Alternatively, I could either use a smarts pattern or a sketcher, and both of these would also allow me to use wildcards and match multiple different cores. If I had even more diverse ligands, I could use the similarity placement. Rather than an exact atom-to-atom correspondence for the placement, it would use similar features to place the ligand in the binding pocket. Now, I have a preview of the ligands that I'm docking, and I can see that this is ligand 1 out of 7. There's a green halo on the substructure that matches my template in the main Mo window. Now, the first couple of ligands, if I browse through here, you'll note they all have the catechol moiety, but they differ slightly in the R groups on the scaffold. However, when I reach ligand number 6, I have a different scaffold for the PDE4 inhibitor, and the same for ligand number 7. This means they don't match my current template and I won't dock them, and I would rather have to use, for instance, the similarity method for placing them. For the refinement, I'm using the rigid receptor. Here I also have an option for induced fit if I want to allow the protein side chains a bit of flexibility during the refinement stage. The final poses are scored with our generalized Bourne implicit solvent scoring function that has an added term for exposed surface area. And then my 3 up to 3 best poses for each docking run has been written to my output database. Now let's take a look at the database. A Mo database opens up in the database viewer. This is the second major window type in Mo. We have the main Mo window for coordinates and the database for looking at the contents of databases. You'll see that it looks a lot like a spreadsheet, but it's actually also holding molecules. So if I expand the cell here, you can see that there's actually molecules in the first field. It's also the main entry point for any kind of cheminformatics and QSAR calculations. Now let's take a look at the data we generated. So as I mentioned, the first field here holds molecules. Those are our docked poses of our ligands. In the field called mSeq, I have the ligand number. So I have ligand 1, 2, 3, 4, and 5, but of course not ligand 6 and 7 because they didn't match the template. In the field S, I have the final docking score. We would like this to be as negative as possible, indicating a stronger affinity. I also have an RMSD score for how much the ligand moved during the refinement stage. The remainder of the database, if I scroll, you'll see that there are energy scores for different stages of the docking run. For today, we're going to focus on the poses. To look at the poses, I'll use the browser. So I'll go to File and Browse, which opens the database browser. The database browser goes through each entry of the database one at a time, displaying the ligand in 2D, but also in the 3D window. Let me move the browser a bit to the side here. Now, the browsed pose is shown in bright green, and what I have here is actually a docked pose of rolipram in bright green, together with the crystallographic pose of rolipram in orange. And what I note is that virtually the poses are the same. So I have recreated the co-crystallized pose of rolipram with my docking run. Now, while I'm looking through my docked poses, I can keep an eye on my browser because there's additional information displayed here. For instance, I have the dock score, the ligand number, and the RMSD refine. I can use the browse keys at the bottom of the browser or the arrow keys on my keyboard to go through the different ligand poses. I'm going through a couple of different poses for ligand number 1. And here I have ligand number 2, which is a different version of the same scaffold, but with different R groups. What I'm interested in is, has my binding mode been maintained? So in other words, is my scaffold next to my glutamine? And do I maintain my hydrogen bond to this glutamine? And I do indeed. So again, this is a ligand that's interesting for me to investigate, and I can have now a look at how does my R group interact with the rest of the protein. Let's continue browsing. Here's a couple of different poses of the same ligand. And now I'm reaching ligand number 3. Again, here I'm looking, do I maintain my binding mode? Do I maintain the placement of the scaffold and the interaction with the glutamine? And yes, how does the rest of my R groups interact with the protein? Let's continue further down. Here we have ligand 4. Again, I'm maintaining my binding mode, and I have different R groups that still fits in the pocket. Here's a couple of different poses of ligand 4 until I reach ligand 5. And again, I'm checking my binding mode and I'm looking at how does the different R groups interact with the protein. Let me close the browser here and bring back the dock panel. So this was template-based docking. It's very useful for docking similar molecules onto a common core when you know the binding mode. In the dock panel, we define the placement by using a superposition on selected atoms, but here we could have also used a smarts pattern or a sketcher. Both of them would have allowed us to use wildcards. If you have more diverse ligands, you can use the similarity method for placement, which would have used a similarity of features rather than an exact atom-to-atom correspondence. I hope you learned a lot today. If you have any questions, please reach out to me at support@chemcom.com.